So the results from the phase 1 and 2a PETRA trial showed that AZD5305, which is a highly potent and highly selective next-generation PARP1 selective inhibitor and trapper, had significantly improved safety, pharmacokinetics, and exposure above target that then could be achieved with the FDA-approved first-generation dual PARP inhibitors. And we showed that 5305 also achieved maximal target engagement and promising clinical activity with a highly favorable safety profile. So in this first-in-class, first-in-human trial, we enrolled and treated 61 patients with advanced breast, ovarian, prostate, or pancreatic cancers bearing germline or somatic deleterious BRCA1 or BRCA2 or PALB2 or RED51C or D mutations with 5305. And the whole rationale behind this trial is that, the approved first-generation PARP inhibitors trap both PARP1 and PARP2 to site of DNA damage, where they then prevent DNA repair and selectively kill cancer cells. However, a growing body of evidence shows that only PARP1 inhibition and trapping is required for synthetic lethality in cancers with homologous recombination repair deficiency, while preclinically PARP2 is essential for erythropoiesis. And therefore, by selectively inhibiting and trapping PARP1, AZD5305 achieved greater anti-tumor efficacy across select tumor and molecular subtypes. It also achieved more durable target inhibition and superior tolerability compared to the first-generation dual PARP1 and 2 inhibitors in preclinical models. And in this trial, the most common adverse events included nausea and mechanism-based hematological toxicities, but at extremely low rates. And ultimately, at the time of data cutoff of this trial, there were no dose-limiting toxicities, treatment-related serious adverse events, or treatment-related discontinuations at all. So none of those. And importantly, only 2 patients required a dose reduction on this clinical trial. And so overall, 5305 was well tolerated with low rates of GI and hematological toxicities compared to the first-generation PARP inhibitors. The excellent pharmacokinetic properties of 5305 were also demonstrated to enable significantly improved pharmacokinetics and exposure above target than could be achieved with first-generation dual PARP1 and 2 inhibitors. Robust and durable pharmacodynamic target engagement was demonstrated across all dose levels by measuring the inhibition of pyrrolation, which showed that 5305 led to maximal target engagement of at least 90%. Of the 40 evaluable patients, 10 achieved RECIST partial responses, 7 which were confirmed, and 11 RECIST stable disease across doses, across tumor types, across mutation subtypes, and were independent of prior PARP inhibitor use. So really, these exciting trials, results of this PARP1 selective inhibitor, 5305, demonstrate that we can build on the success of the approved first-generation dual PARP1 and 2 inhibitors, and we have provided important clinical proof of concept for this innovative strategy in achieving substantially improved safety, pharmacokinetics, pharmacodynamics, and promising efficacy in patients with different molecularly driven cancers. We're now conducting expansion trials to expand and to evaluate the drug's efficacy in PARP inhibitor naive populations, and dose expansions and also dose escalations of combination therapies, including trastuzumab deruxtecan and datopotamab deruxtecan.